I think the screen is visible.
Yes, sir.
Anybody? Yeah, okay, fine. So as you see in the instrument, it's all about the LCMS. So LC is one part. LC we discussed yesterday. There are different detectors of the LC, and one of the detector is MS. So in a simple language, it's one of the simple detector, but we will cover what is inside the simple detector as such. So in today's session, this is my agenda. So we will include what is MS, why MS we are using, or what is the purpose of MS, and how it works. Apart from that, now what we need to do, or what are the different applications or types of MS that includes either HRMS or a hybrid, or maybe that it includes the TOF or MALDI or different types of MS, and how we can achieve our target by using this detector. And obviously the session is open for the further discussion about the MSMS if you have any concerns in your mind. So I will try to finish this session within the next 85 to 90 minutes, before 90 probably, because I required 5 minutes for just question and answers at least. So it's a very elaborative session. So have a patience up to 85 minutes. Okay, so this is the simple definition in my terms. So when we are using different instruments, normally terminology, being as a chromatographer. So as a chromatographer, you must know the chemistry of any kind of sample. The first one is, if you can evaporate, GC can separate, is a simple terminology. If sample, whatever sample you have, if you are going to evaporate it, obviously GC will separate it. In LC, if you are able to dissolve, LC can resolve in simple terms. And the last is the most important regarding the LCMS, and that is if you can ionize, MS can recognize. So how ionization process has been takes place? What are the different terms in the spectroscopy? What is the starting point of MS? So before going ahead, I just want to tell you each and everyone about this mass spectroscopy is a universal ion detector. It's not a universal detector, by the way. A lot of people also telling me in some norms that it's a universal detector, but ELSD is a universal detector, and mass spectrometer is a universal ion detector. So I'm not going in detail about the definitions and all those parameters, because you all are currently doing all the definitions, and I have a lot of exams over there. So I'm not focusing anything on the definitions. I will give you the practical demonstration, how it works, what are the different phenomena of the MS. So with starting, if you are talking about the LC, as I explained yesterday, in LC you will find one dimensional information, like if I plot a graph of time against the intensity. So if there are different indicates or different peak, you may observe over there, depend on the retention time, and you are able to specify the presence or absence of that particular compound. So it's a one dimensional. But when it comes to the MS, it gives you the second dimension, that is molecular weight as well, apart from RT. So it indicates the presence of a compound or the specific compound with a RT as well as with a molecular weight. So we call it as LCMS, is a second dimensional. But the third one, that is LCMSMS. So what is LCMSMS? It gives you the retention time information with the help of LC and molecular weight, as well as the fragmentation pattern. That is, what are the different fragmentation pattern of that particular compound. So I just attended the last session as well, almost last 40 or maybe 30 minutes I was there when Marathe sir is explaining a detail about the structural isolation, and I was very happy that I mean that same field I'm doing the NMR and preparative HPLC and LCMS as well for the structural isolation, and that was very good information. So regarding this fragmentation pattern, when you are able to identify the fragmentation of each and every component, it's very useful for you Just like in IR, it's a fingerprint region. So just like in MS, it's a fingerprint region for each and every fragment. So fragmentation pattern is different. There are n number of molecules in the universe showing the same masses, but each and every molecule has its own different fragmentation pattern depending upon which molecule attached to which position, at what position, and what are the different compositions, or what are the different functional group attached to that component. So we are also discussing on the MSMS part. Before going ahead, we need to understand, yesterday I think one of the sir asked me regarding how, what is actually interface, how MS or HPLC sample goes into the MS, and what are the different technique used for that MS. And for that purpose, ionization terminology is playing a most important role in the kind of MS. I'm talking about LCMS. So it's called as atmospheric pressure ionization. In the atmospheric pressure, there are two different ionization process. One is, or you may say, sources. So the first source is ESI, that is electrospray, and second one is APCI, atmospheric pressure chemical ionization. So these are the two main sources. Apart from that, APPI is also there, but we will focus on ESI and APCI according to the market trend. So ESI, normally the main difference of the ESI and APCI is when the technique has been formed, the solution droplet, when there's a formation of droplet at the same time, ions are formed, and then that droplet are evaporated. So ion formation is the most important criteria. So whenever there is a formation of droplet, at the same time, that droplet get ionized. In APCI is something different. So at first, this technique where analyte and mobile phase is first evaporate and then ionize. So we are just applying the charges before formation of droplet or during formation of droplet, and in second case after formation of droplet we are giving some charges to that particular ion. Why is it so? How it works? Let's see. So I'm just taking the example of a simple molecule of a caffeine. So if you want to analyze a caffeine molecule, or you want to say that information about the mass of that caffeine, so just a moment, I will use the print just pointer. Okay, laser pointer. So this is the structure of structural formula of a caffeine. So I'm not going in detail about the how you are going to calculate it, but yeah, as the total number of nitrogen, total number of oxygen, and obviously C8, H10, N4O2. So you must know the molecular weight depend on this periodic table. By using the periodic table, this is very easy to identify the molecular weight or using the molecular formula. So depend on the number of elements present in that particular moiety, we need to define the total weight of that mass of a carbon. Likewise, 10 into 110, then n nitrogen are 4 in number, so we need to calculate by into doing the 14 into 4, 56, and 2 oxygens, so it's 16 into 2, 32. So total molecular weight is 194. The caffeine molecular weight is 194. When we are talking about LCMS, either it may get protonated or deprotonated. So either you will get the mass of either in the form of M plus H or M minus H. Okay, so the caffeine molecular weight either you will get as 195 or 193. So in simple language, we are just doing the weighing of your molecule. So molecular scale is all about LCMS. Okay, then why MS over the other detector? Obviously, yesterday I also just having given you the some of the glimpse, like unmatched sensitivity, higher detection limit, that is in the terms of picogram, or you may say parts per trillion. Now it's not parts per trillion, it's going beyond that, pictogram as well, in different depend on the different sensitivity limits. and different detectors we need to use in the LCMS. Speed and diversification of the application. So it's a high speed instrument. You may get the results within hardly 1 minute or maybe 3 minutes. So it's as good as fast, and it's a universal ion detector. Yeah, it's a simple, but it's not easy one to, you may say in the terms of handling or maintaining. But if you know how to handle the instruments, it's very easy or a very useful instrument for structure elucidation. It will suffice your almost 30% work. NMR also suffice your 30% work, and you will get the identification extra. So in before going ahead, there are some concept I want to clear, have. I just want to introduce some concept, almost 4 to 5 concepts are there. It is how mass spectrometer has been used. So normally mass spectrometer is a powerful analytic techniques that can be used for identification of an unknown molecule like impurity profiling, whatever we are doing in the structure elucidation. So those are the unknown molecules. If I'm going to purify that molecule, say example, I have one API with me, I need to purify that API, and the percentage or the concentration of the API is almost in the, if you see, if you are calculating by area percent, the calculation is maybe 80%. So I need to find out what are the remaining 20% is all about. Either it's an inorganic component, either it's some impurities present in the sample, or need to identify it. So I need to develop the analytical method for the better separation, and then I will go for the purification using the prep HPLC or other techniques as well, so as to get that pure compound in my hand. And after getting that sample or after getting that impurity, I need to evaporate using the lyophilizer or different techniques. And after lyophilization, we will do either carbon, CMA, proton, or maybe IR, or along with that, we need to perform the LCMS. In LCMS, there are 2 techniques. We need to just identify the molecular weight, or we need to focus on the other part, that is fragmentation. So this will help you to identify the structure or elucidation, as well as checking the chemical properties. So yeah, it's a powerful compared to what quantify and elucidation, as already I explained you. Then a simple definition is, you must identify the molecule present in solid, liquid, or a gas sample. If it is in a gas, obviously we need to use GCMS. Determine of the quantity of a molecule, obviously we need to quantify if it is required. So quantification is one of the other tool which is very important while you are using LCMS, because qualitative analysis is not that much difficult when you are just identifying the sample. But when I need to quantify how much compound present in that particular matrix, then obviously MS will help you a lot. So this is a first concept. Second concept is sorting and counting. So I'm just showing one of the picture of a cent or maybe quarter or the maybe dollars. So they are all mixed together. I need to sort it out, and after sorting, how much dime are present, or how much quarters are there, or how much dollar is there, nickel and half dollar is there. I need, it's like, in Indian currency, you may say it's a paisa, rupees, and all those things. So we need to identify how much quantity is there, and depend on whole calculation, it's very easy to calculate the total molecular weight. So what is MS and what is the MS interference? So everybody knows about HPLC. So HPLC, I explained yesterday, it contains aqueous as well as organic solvent as a buffer. So either you are using some buffers like some acidic buffer like TFA, or maybe sometimes formic acid, or maybe either acetic acid. So those are the acidic buffers in the percentage of hardly 0.1% or maybe 0.05%. So if you compare towards the basicity, so it's like ammonium acetate buffer, ammonia buffer, or maybe some of the phosphate buffer. But there are some do's and don'ts while we are using LCMS. Normally, phosphate buffers are not allowed. TF is also not allowed as a buffer. The reason behind it is that phosphate buffer will form one salt and it get deposited on the detector, and it's very difficult to remove that salt present over there. And then obviously we are getting a very less sensitivity after some time. So we are not using phosphate buffer. It's very difficult to remove the phosphate buffer on MS. TFA normally we are not using because TFA is showing the mass of 113 in the negative side. It's a very strong ionization of a TFA, as it is a trifluoroacetic acid. So it's showing normally acids are obviously showing a proton donor, so it's CO minus, and that's why it comes in negative. So 113 is a higher response mass, and that hindered the other masses. So we are not preferring to use TFA as a buffer. So for that purpose, we required something in between when it's we are passing this HPLC waste as a MS inlet, so as to it gets ionized properly. So we called as interface. What it does, so interface is a most and important phenomena whenever you are using any kind of MS. So what it does basically, it's a core technology. It's removes solvent. It is first role to remove the solvent, and second one is ionize the analyte. So these are the two important phenomena we need to perform in interface before going in actual MS. Whenever the sample you are injecting from HPLC to MS, interface removes all your solvent and it will ionize the analyte as well. So as I just discussed, there are different sources. So all sources are placed over here. APCI or EFCI is placed in interface. So how it works. And the third concept is ion and charge ratio. So whole LCMS concept is based on ion and charge ratio. Everybody knows about the ion, how ion and charge ratio is there, why we call it as M by Z, because either you need positive charge or a negative charge to detect on MS, at least on LCMS. As today we are discussing on LCMS. So the ion is electrically charged molecule, and obviously either it carries positive or negative charge, and molecule must be charged to measure on LCMS. Okay, so the mass spectroscopy weighs molecules electronically by attracting and repelling ions. So as concept, it's self-explanatory that it should be either positive or negative charged. Concept number four is ionization technique. How ionization technique helps to make that proton either in charged or deprotonation or a protonation. So formation of charged liquid droplets from which ion are wiped out. So this is the simple definition. If we want to say, it's an electrospray. Electrospray means ionization of a process of charging the molecules. So we are just charging it by applying some electric charges. So how we are applying electric charges? No, we'll definitely let you know in coming slides. So concept number 5 is obviously tandem mass spectroscopy. So MS1 is only the sorting a molecule. Then fragmentation chamber, it's a breaking the molecule. And MS2, that is, this is about the triple quadruple. So MS1 is just identifying it, molecular weight of a compound. MS2, that indicates it we called as a fragmentation chamber, where actually fragmentation happens in Q2. So it's a breaking the molecule. And third one is sorting the pieces. So we need to sort out the piece corresponds to which molecule. So if this peak is there or if the fragment is there, this fragment corresponds to which molecule we need to identify. So these are the some concept, basic concept before moving ahead. So there are different types of mass spectroscopy. Quadruple, the name itself explains there are 4 quadra. A quadra means 4, and 4 poles are there. Time of flight we call as TOF, ion trap, magnetic sector, then IT TOF. So these are some of the MS. So we will see what are the different techniques and which MS is more preferable as per the applications. So quadruple, ion trap, and TOF. So if we are qualification for unknown sample, quadruple plays a huge role. It's a more important rather than ion trap or TOF. When you are talking about selectivity and sensitivity or quantification, quadruple once again shows you the higher selectivity and higher sensitivity. So in terms of all those techniques, obviously each and every technique has its own benefits, but in general terms, if you want to qualify and quantify, this is the most important LCMS, which is called as quadruple. So how it works basically? Before that, quadra means 4 poles. So I'm just placing one of the diagrams showing the 4 different poles, and detector is here. The mixture of component is here. There are 4 components present in a mixture. So what I will do, just passing that mixture through the quadruple and see the result. So after passing those 4 different mixture, each and every component gives his own spectra, own data corresponds to the depend on the structure. So we are able to identify the molecular weight of each and every component separately. And during this, the feature is it's used for general purpose, most widely used for scanning purpose, or maybe the SIM. So what is SIM? What is MRM? I will let you know in coming slide. But the advantage of this is it's a compact and lightweight, easy to operate, high selectivity and high sensitivity for quantification as well. And that's why each and everyone prefer to have a quadruple at least. So this is a single quadruple where it's very difficult to get the fragmentation, but very easy to get the qualitative analysis. Now, talking about time of flight, we call it as TOF. What TOF does basically, it's good for quantification. It's measure time taken from the starting of this tube to the end of this tube, the time taken to travel that ion towards the detector, and it's measure that time, and it's good, normally commonly used compared to the comparatively it's obviously it's a comparatively large as you see in the MS, but it's it is useful to just scan the data. Advantage is obviously it is a high resolution, and high spectral sensitivity is also there. But the main disadvantage is that it's you may say quantification is not possible with the help of TOF. So what TOF does, so it giving a 3 or maybe not 3, it giving n number of spectra, because here's a one filter, we are applying sample from here. As sample come, it's not about sample. If we are talking about MS, we need to talk in the ions. So when ions will come over here, the mixture of ions, so obviously depend on the you may say the density or the bulkiness, the time taken to pass from this tube, it shows the masses of a different component, it's showing the resolution. And obviously it requires high vacuum, it has some disadvantage, it's bulky in nature, so quantification is also not possible in TOM. There are other principle we called as the ion trap. So what is ion trap is actually, it is a good tool for quantification, as it gives you the we can trap that particular ion if you know the molecular weights. Example, mass is 501, 501 is your molecular weight, and I need to trap 250 ion over here in the trap. So 250, obviously it's the fragment of 500 probably. I'm just guessing the fragmentation pattern. So it is a comparatively small, very easy to use. It permits to scan the sample. So scanning, everybody knows about the scanning, right? I'm just scanning my body, that indicates I'm just identifying what are the different parameters present in my complete whole body. So same, I'm just scanning the sample. If I inject the sample for the scan mode, it will show you how many different molecule present in, or how many different masses are there in that mixture. So it's just simple scanning. Advantage, obviously it gives you me the proper identification. So as you see, I'm just trapping it and just allowing one by one so as to move ahead. Obviously it is a disadvantage of those ion trap is that quantification is not good. Normally nobody is using for quantification. Poor resolution as compared to quadruple, and obviously we have some limitations in MS due to the limit on the trapped quantity. So coming to the mainstream, that is the quadruple. So combination, how it works. So now it's we called as SQ, that is single quadruple. And single quadruple means here your HPLC is here, either GC or HPLC, and ionization will happens with the help of 4 quadruple and is a mass separation. That's it. And obviously detection. So only 4 quadruples are present in single quadruple, that is we called as SQ. It also contains ES and APCI, and in GC it contains EI and CI, chemical or either electrical. So in Q quadruple, there are mainly with the help of either TOF or maybe single quadruple is there. And about the triple quadruple, we need to club one more quadruple and then detector. In between these two quadruple, there is one different chamber we called as QRA. Normally it's a Q2, and where the actual fragmentation takes place. So Q2 plays a huge role in the triple quadruple. And in this scenario of the LCMS, so today we are just focusing on LCMS, so I'm just comparing the LCMS. Either you are using single or triple quadruple, this ionization chamber also remain the same, either with the help of ESI and APCI. And obviously there are different type of the masses present in the LCMS with the different quadruples. And the ranges from the model are 8040 to 8050, 60, like that. Depend on the sensitivity, you may select, or depend on the your application, you have to select the different models. So essential of the LCMS, the first and foremost part is ionization. Why I'm focusing this part? Because I'm taking almost 80% of all today's presentation focused on this first part, because this is the most important parameter before going to the MS. So ionization is the most important part. Either it's in the terms of ESI, that is electrospray, APCI, DOS. So DOS means it's a combination of both ESI and APCI. We call it as DOS. If you want to identify the polar, mid-polar, or the non-polar component on the same pattern or on same run, the DOS is useful. The only disadvantage of DOS is it is less sensitive as compared to these two. So, and APPI as well, atmospheric pressure photon correct. So normally we are using a carrier as a vacuum, then separation as either in the with the help of quadruple or drop, or maybe the trap. And detectors are normally we are focusing on EMT or PMT or ECM. So how it works and what are the different components, or how we need to target our molecule, in which, or either we need to use ESI, either we need to use APCI, how you are going to identify. Say example, your molecular weight is highly polar, or molecular weight is showing, that's fine. If it is 1000 or maybe 1500, that's fine, or maybe it is a 400, but depend on the polarity. Okay, normally we are choosing the first one is the polarity, second one is volatility, third one is depend on the large molecular size, and is that molecule is thermally stable, or how it behaves with the temperature. So that plays a huge role. So I'm just plotting the graph of molecular weight against the polarity. So it's a non-polar moiety over here, and then medium polar and high polar. So high polar to mid polar section is all covered by ESI, high polarity. Then towards the non-polar component, we play with APCI, low or medium polar. And then in come the picture with if it is a volatile, then obviously we are focusing on GCMS. Okay, so this is the area of ionization basically we are focusing on. So if we compare PDA versus LCMS, so PDA means yesterday I shown you different types of the graph obtained with the help of the depend on the wavelength. So the red one is your chromatogram. Just see over here, red one is your chromatogram showing the two different peaks, and each peak corresponds to two different wavelength. So, or the first one, so one is obtained at 4 maybe 580 nanometer, second one is 210. So these are the two different wavelength. And if the LCMS come in the picture, you will get the idea of the two different compound as well as the masses of those two different compounds. So it is an added advantage if you have PDA plus LCMS when basically in the research lab or the lab where we are just focusing on the unknown samples. That will help you a lot. So how it configuration? What is the configuration of MS? So yesterday we discussed about the mobile phase, degasser, then solvent delivery module, that's we called as pump, then the mixture where it's mixer we get the mobile phase get mixed over here, and then the autosampler where we are putting the sample. I'm just explaining the flow diagram. After autosampler, it comes with a column 1 where we kept a column with a specific temperature, and after separation of the after separation on column, each and every compound or each and every API or maybe the unknown impurity passes into the detector. First one is either PDA or UV detector, where we are getting some signals, and then either it goes to the waste in the part of LC, but in the part of LCMS, it works as the input for MS. Okay, so in the gases, what are the different gases we are focusing on? We require gas for the nebulization and for a drying purpose as well. So we are using nitrogen gas as nebulization gas, as a drying gas. So here we are using for the heating gas. Why heating gas is required and what is the importance of the collision gas, why argon is used, we'll explain in detail in a brief in coming slides. So we will talking about the ionization of a molecule. This is the ionization probe. When sample come from the after the separation, it comes to the MS. Either it get protonated to form the positive ion charge like this, or it get deprotonated to form the negative ion. Okay, that wholly depend on the structure of your molecule. So you may find the section or dissection if you see the CTs of the MS. So this is the ionization probe, and we are placing a DL line. We call it as a dissolution line. So up to this ionization probe, there is atmospheric pressure. We are not required any vacuum as such to implement over here. But as the starting from Q0 to Q3, we required certain vacuum. So here we are normally called as Q0. What is Q0? This is the Q0 where we are normally we are focusing on the QRA to smoothen or to focused on the masses. So to pass into Q1. So the Q1 is a single separating unit, Q2 is a collision cell, and Q3 where we are getting the fragmentation pattern, and obviously the last one is a detector. So this is the UMSA. It is completely under vacuum. This part is not required any kind of vacuum as such. Now, if you are focusing detailed about the MS, so I just make one how it construct basically. So we required this is called as ESI. Okay, any probe, either ESI or APCI. So mobile phase coming from the HPLC, we need to put it over here. After putting this mobile phase over here, there is one capillary inside this ESI. We called as ESI capillary, having the dimension of hardly 0.3 mm of ID, not more than that. So along with that capillary, there is one gas we call as nebulization gas. It passes from the source of ESI as well, and it forms the sprinklers. You may call as nebulization happen over here. Okay, when a drop come from the mobile phase, that drop will form sprinklers, and we are applying certain temperature in the ionization probe. That's why all your mobile phase get evaporated, as your mobile phase contains hardly water or your organic solvent, either methanol or either acetonitrile or methanol. That's it. So all get evaporated, and then the part of DL line, so it's a dissolution line is there, so it come in the picture, And because of high vacuum, all ion are stuck inside this DL line. And after that, we are using QRA to channelize that whole ion in a single channel, and where we are using UF lenses. This lens helps to synchronize the channel of the mass in a one single pattern, and then come in the picture with a Q1 and then Q2 and then Q3. So in detail, what are the different part required in MS? First one is sensitivity, second one is selectivity, and third one is also selectivity. So sensitivity is the most important part of any kind of MS. Okay, so this is all the flow what I explained you in detail. So first of all, just focus on the ionization. So we are using heated ionization probe. So heated ionization probe means what? Whenever the mobile phase is coming from this capillary, apart from that, there is one blue line, normally we call it as nebulization gas. So as a droplet will come over here, nebulization gas help that droplet to form the nebulization. Okay, it forms different droplets, and that droplet get converted or evaporated completely, and only ions are passing into the MS. So that's why we are using heated ESI probe, where we are just heating the air, and that air is also passing along with that droplet. So see the example, the sensitivity goes on increasing. If we are not using heating gas, the sensitivity is like this, and if you are using heating, not using and using, so heating gas is off, sensitivity is low, heating gas is on, sensitivity is almost 30 times more than the normal one. So depend on the your ionization, we are putting different modes. So the second one is dissolution line, this one. So ionization probe and then the dissolution line, we call it as a DL line. How it helps? DL line is a just channel to pass that ion into the MS interface. It's a first source to introduce the ions for the MS. And when DL line get blocked, we need to remove it and wash it thoroughly, and just once again need to put it. But normally what happens that whenever you are removing the DL line, normally vacuum gets dropped. But in certain levels, we are using the orifice over here. So orifice will introduce directly when you are removing DL line, and that orifice avoid vacuum trap as such. So this is the DL line. This green color is your DL line mixing. It's so now coming to the main part, basics of the terminology. Now you will get the fair idea about the ESI and APCI. So whenever the sample is coming from the HPLC as a drop liquid sample, first one is a drop, then with the help of nitrogen gas, it will be called as the nebulization gas, it get nebulized, there are different droplets, and then during the droplets we are charging the different voltage, almost 3 to 5 kilovolt is there, 3 to 5 kilovolt is there. So normally that kilovoltage will help us to form the ions. So during formation of droplet, we are introduced the charge voltage, and that's why charge has been formed, either positive or negative, depend on the proton donor or acceptor. In second, that's why we call it as a soft ionization technique. By the way, ESI is a very soft ionization technique and widely used for the highly polar and mid-polar samples. What happens with ESI? Not ESI, it's APCI. So first of all, same technique, same sample coming from the HPLC. Apart from that, nitrogen gas is there, but here we are not applying any charge as such. At first, it get evaporated, mobile fluid gets evaporated, nebulized, and then forms the droplets. And after formation of droplets, that or maybe the sprinklers or the ions, those ions are bombarded on the needle, we call it as corona needle. So this is not a corona which is coming in 2019-20. This is almost old corona needle, which is there since last 40, 50 years. So that corona needle, we are normally have implemented the high voltage, okay, 3 to 5 kilovolt voltage, and with that voltage, it either get protonated or deprotonated. Why is it so? Because we need to implement, because it's as it is a non-polar in nature, it is a very rare to get ionized. So we need to break that ion into the small ions, and then we need to apply the charge. And that's why we called as the hard ionization technique as APCI. So the basic difference between ESI and APCI is a soft ionization. Second one is a hard ionization. During formation of spray, we are implementing the voltage. After formation of the ions, we are implementing the voltage. And if called in the regards with the DOS, obviously both are doing simultaneously. We have this probe as well as the corona needle inside the DOS. Now, in detail about the MS, same whatever I explained, the flow rate from the HPLC is 0.01 to 1 ml. Normally, we are not allowed to pass hardly not more than 0.5 ml flow per minute inside the LC MS. This is the nebulization gas. So it forms the spray with the droplets, and then as we are applying a certain temperature of that ionization probe, due to the temperature, that droplet get shrink, and as the surface tension increases, it forms the n number of droplets. So we call it as a declusterization in simple language. So it forms the clusters, and then obviously more major clusters. So either it gets M plus H or M minus H, and then that ion passes into the DL line. But during this DL line, one more drying gas is there, which is applying the opposite force from the MS. So that why we are applying drying gas from both sides to dry that ion properly, and then only that ion passes in the vacuum. So all this is happen in ESI probe. Interesting, right? So this is all about the Coulomb explosion. Normally we are using the analyte declusterization, whatever I explained. It's a solvent ion forms and analyte ion forms. So because of the evaporation, it gets Coulomb explosion, and then explosion it forms the solvent ion as a cluster, and once again it's a completely get declusterization with the help of same phase. But whenever you are talking about the APCI, liquid sample apart from nebulization gas, then heater, and then charged, and then formation of ion molecule. So this is the vaporization. So whenever the analyte combination, this is called, we call as aerosol. As it is not charged, we are not calling any charged ion, so it's a complete aerosol. After getting the evaporation with the different temperature, normally the temperature is 200 or 250 maximum, then it vaporized completely, and then it come in contact with corona needle and get charged. So there is a formation of analyte. So this is the first phenomenon. Now, how we get configuration? So this is we called as probe. Everything is happens over here. Whatever I explained till now is every parameter applied only for this region. Okay, either this or this, maybe either single chord or maybe triple chord. So the lenses has come in the picture, which allows you to narrow the length, because once again, coming from the capillary, the ions are in the form of spray, right? So we need to channelize those ions. So for that purpose, we require a skimmer, it having a dimension of hardly 0.8 mm. So imagine 0.8 mm skimmer is there. So then whatever the masses are there, it gets completely channelized and then passes towards the Q2, Q1 and detector. So this is in the case of single quadruple. If it is a case of triple quadruple, obviously the thing is same, only the skimmers are increased so as to get the more channelized. So number of ions are here in this region. We normally, this region we called as Q0. Okay, so as to get channelized, we are applying some skimmer and then mass pass from the skimmer towards the Q1. So obviously it's a quadruple. So in the picture, it's not possible to show the four poles. That's why I'm just focusing on two. But either in the Q1, then collision, and then Q3. So yeah, these are the three different stages. So then why required a vacuum in such scenario? So to provide the free path for the ion, vacuum is must. And how much vacuum, or what is the level of a vacuum? It's almost e raised to -7 torr. It's a very high vacuum. So I also come in that vacuum stage. Why is vacuum is required? Mass spectrometer must function under the vacuum. This is necessary to allow ions to reach the detector without undergoing any collision in between. So when the mass is passing from this spray of DL line, so this is your dissolution line. So this is atmospheric pressure. We don't require any vacuum. From here onwards, we require some vacuum. So this is a lower vacuum and this is a high vacuum. So lower vacuum will be taken care by the rotary pump. Rotary pump normally we are applying for, we are called as a roughing pump, you may say. So this roughing pump will apply to have some vacuum, at least 5 Pascal or like that. So the we applied maybe 100 to 200 Pascal as a minimum vacuum. And then from here, as a thermomolecular pump, we are applying the different pressures. And the if in the terms of Pascal, it's hardly 3 into raised to -3 Pascal or less, depend on the CID gas. So what is collision gas? Obviously, collision gas is your inert gas, and that's why we are using argon. And if you see the size of this skimmer is 0.8 mm in ID. Okay, so RF voltage and DC voltage also plays a very innovative role while moving the charged or the mass ion from the quadruple. So if you see from the front, these are the quadruples 1, 2, 3, 4, and from the above you find these are the four quadruples. So the picture I put it in a different ion having the different colors. Normally it's a green or maybe the red or maybe the blue. So each and every ion is passing over here. We need to optimize the RF and DC voltage. Okay, and depend on which molecule I am interested for. For example, if I am interested for this red color molecule, I need to optimize the voltage of this rod. We call it as pre-rod and main rod bias. We need to optimize the voltage, and depending on that voltage, that goes towards the detector, and rest of the sample or rest of the molecules are vanished before the detector itself. So the use of those quadruples are opposite poles carrying the same charge. If, for example, these two are having positive, these two are having negative, and it change the, you may say, the charge change within a fraction. We normally in the current scenario it changes in 5 milliseconds. So 5 milliseconds the changeover of the you may say charges. So it's a drastic change. If I applied positive charge over here, for example, this rod is having positive charge and my molecule is passing from this halo region, and this molecule contains negative charge, automatically this will attract towards this quadruple. Okay, because it's having the positive charge. But on the spot, in a hardly in millisecond, the charge will get reflected as a negative. Then it repelled and attracted towards this. Then once again attracted towards this. So all momentum is continuously, it's a random moment because the charge changes fastly, and that's why it's very easy to move ahead for that molecule. Okay, same explained the quadruple theory over here, just applying the concept RF and DC voltage. So this is all about the quadruple theory. Now we are focusing on how we analytical modes are important and how we are getting identified the analytical different modes. So when we are focusing on the analytical mode, we have the different modes like scan, SIM mode, product ion scan, precursor ion scan, MRM, and neutral ion loss. So don't get confused. Each and every segment or each and every mode has its own specification. For example, scan means I'm just scanning the sample, nothing else, passing through Q1 or Q3. just qualitative analysis of the structural elucidation analysis. So whenever I am focusing on the SIM mode, that is we called as single ion monitoring. SIM means single ion monitoring. So in single ion monitoring, I'm just passing as a single mass, passing in Q1 and Q2 for the quantitative analysis. Then product ion molecule and precursor ion molecule. So precursor means, as I've taken the example of caffeine, so it's a 194. If it is ionized in positive mode, 195 is my precursor. Okay, and the fragment of that caffeine is maybe 100. So 100 is my product. So precursor means your parent molecule, and product, that means it's a daughter one. So parent and daughter. Normally, we in a simple terminology we may call it these loans. So precursor is in a scan mode and SIM mode. So MRM will show the SIM mode in both. We will see each and every part in very you may say different modes. So how the scan mode is going to take place with Q1 and Q2. Normally it's scanning from the different modes. Example, we have the different masses. So it scans like this. So product ion scan if I'm focusing on only single product, or precursor ion if I'm focusing on different precursors. So it's better to explain in this way. So example, the mass is the sample mass carrying 500, 300 and 200 probably. So in scan mode, I am getting each and everything. So I'm getting the higher mass, I'm getting this fragment as well, and as well as the other fragmentation pattern. So, but in the product ion scan, if I am focusing on a single molecule, like 603 is a molecular weight, and if I am focusing on only on 300, so that 300 passes on the Q2, which is called as collision cell, and in the collision cell, the fragmentation CID gas has been applied. So collision CID gas means collision energy. So we applied some gas to form the fragmentation, and that gas should be inert gas. So we are applying argon. So we call it as collision gas. So normally argon is the best source because it's inert, and we get the fragmentation of mass of 300 in the form of either 200, 150 or 100 or like that, and same can be passing through the Q3, and I'm just getting the mass of 300 and the fragment of 300 which is corresponds to 300. That's it, nothing else. So there are two different experiments. Either we need to just scan it, or we need to focus on single moiety and focus on that fragment only. So what I will do, I think it will be better to show you a systematic way. Just a moment. Yeah. So if I am doing a product ion scan, in simple way, if I am doing a product ion scan, that means I am focusing on the molecular weight of 500, okay, 300 and 200. When argon gas is applied, that means when I am doing fragmentation in Q2 and the mass range is from 10 to 300, what happens over here? I'm just focusing on 300, mass of 300. So mass 300 will pass from here. So 500 and 200 will not allowed to pass in Q2. Okay. And only collision, after applying collision cell, there are some fragmentation pattern observed in Q3 and get detected. And I'm getting this type of spectra. I'm getting the mass of 200, 150, or maybe lower mass as well, maybe 20, 30 or like that. So this is the product ion. Now in the precursor ion, I'm allowing to pass each and every molecule or each and every ion to the Q2, but I am just focusing on, I'm just filtering to pass 150 in Q3. So 150 either corresponds to 300 or either corresponds to 500. We don't know, right? So there are two options, either precursor or product ion. So in precursor, we are focusing on molecular weight of 150, which either corresponds to both, like this. For example, all ions are getting formed and I am getting the response of 150. But the response is almost double as compared to the 150 single mass. And that's why we are checking this analyte either corresponds to 500 and 300 as well. So we need to cross check it, and that's why precursor ion will help you to check the what are the product actually. Okay, then the next one is a neutral ion loss. Sometimes some of the molecules have a tendency to lose the water or lose the nitrous compounds or lose some of about the sulfur. So those molecules are fixed almost -32 or -18 or -41, or maybe it's depend on the structure. So the mass is coming. For example, I'm focusing on mass of 500, but I'm always getting a mass of 400. That means something is missing. So loss of 50 is there throughout the analysis. So in that scenario, there is one option we call as neutral ion loss, neutral loss scan. So I know that mass is less than 50 in each and every fragment. So what I will do, I'm just passing all masses from Q2, okay, and all masses are getting reflected in Q3, which is showing either the mass of 450 and 250, which corresponds to either 500 or 300. So if I use this loss of ion, obviously in whole scenario, this neutral ion loss is very rare. Hardly we are using in some percentage, maybe 5% or 10%, not more than that. But yeah, this is also important phenomena. We need to implement it. Obviously, it corresponds to 500 and 300. So there are different models depend on the selectivity and sensitivity. So as you goes on increasing, the sensitivity also goes on increasing. It's whole depend on the your applications. So there are all different phenomena normally. As sensitivity is higher, you are getting better results, but obviously you are getting the other options as well. So depend on the scanning speed, your mass. When you are focusing on mass, there are two important parameters. One is the scanning speed, and second one is polarity switching time. As I mentioned already, its polarity switching time is 5 milliseconds, and scanning speed is a 30,000 Dalton per second. So per second, it actually implemented the 30,000 Dalton scanning. Okay, fine. So before moving ahead, uh, does anybody has questions about the LCMS? So I will cover the part of the applications in detail. So I think I will stop this screen and let me know if somebody has a question about the LCMS, because uh, on application point of view, we will discuss in detail about the applications. But till now, somebody want to know anything or having some doubt? Hello, am I audible?
Yes, sir, you're audible.
Yeah, okay. Nobody has any doubts. That's why I'm just
Dear
asking.
participants, if you have any questions, please ask to sir.
About the instrumentation part or
Yeah.
about the techniques, different techniques. Not a single doubt on MS.
No, sir, you can continue, sir.
Okay, fine. So what I will do, I will cover the application part. So main part, applications. Yeah. Normally, LCMS applications are in all touching all the peripheries, starting from the clinical trials, forensic chemistry, food and safety analysis. Now it's booming like anything. Pharma sector, you know very well that in the pharmaceutical, including API as well as after formulation as well, normally nitrosamine is playing a huge role. So pharmaceutical industry as well, environmental science. So normally we are using for the pesticide analysis, life science, normally protein analysis, or maybe some biomolecules, biosimilars molecules like peptides, or maybe some amino acid analysis. So it's a huge applications database is there. Almost, I just gone through some of the article where I found that using LCMS, they are using no the application number wise. If you say forget about the these different domain, but if we see the applications about the domain itself, there are more than 20 to 30 domains are there. Okay, and in each and every domain they have almost n number of articles, n number of publications, so and different applications. So normally residual pesticide analysis, veterinary drug analysis, water quality analysis, or maybe drug of abuse, rapid toxicology is now playing a huge role, narco test or like that. So metabolism also playing a now, newborn screening is coming the picture since last. It is compulsory in the outside the India in foreign countries, but in India still it's under process. So obviously it will come in the a huge role as a metabolic studies. Whatever the testing, blood testing you are doing, calcium content test, or either cholesterol, everything is done on the LCMS. So if you are submitting the sample for thyroid care or any other pathology, the almost 70 to 80% of the testing has been done with the help of the LCMS. So cell culture profiling, metabolic enzyme in the yeast, or maybe phospholipid profiling. So all are the different phenomena. So I'm just putting some of the applications. While just explaining the different applications, I'm focusing on some amino acids. So simultaneous analysis of amino acids. Normally, amino acid analysis on the HPLC is very critical part. We need to do the derivatization, as amino acids are not active, UV active. First of all, we need to activate as using the fluorescence and derivatization, and after derivatization either it's visible on lower wavelength, or maybe on the higher side we need to quantify it either using the pre-column derivatization or post-column derivatization on the HPLC, so using the fluorescence detector. But if we are using LCMS, we are not doing error derivation. It is without derivation. We just need to quantify it or maybe qualify it, depend on the molecular weight of a compound. So that's also possible. One of the application just we covered, as this is on the model 8045. So we covered almost 836 pesticides in hardly 11-minute run. Okay, so this is the important criteria. Within 11 or 12 minutes, I'm able to identify almost more than 800 pesticides. So the question comes, why 800 pesticides are required? Now it's a grape season, right? So now everybody knows about the grapes. It's very tasty, but apart from that, it contains high pesticide. It's more than 400 and 400 or 500 pesticides are there, while different pesticides used while grooming them. So we need to quantify it. We have some MRL level or maybe some LOQ level for those analysis. We worked with the different veterinary drugs as well, which help us to some of the different pesticides in veterinary. Melamine content in the milk is one of the crucial phenomena. So melamine is also a very important factor. We need to find out in the parts per billion or maybe parts per trillion figure. We need to go for the LCMS analysis. So, and in the pharma sector, mainly to identify, elucidate the structure, or to get the proper idea of some nitrosamine, some carcinogenic compound. So nowadays, I think everybody aware about the US FDA norms. So everybody focusing on the nitrosamine impurity, as it's a highly carcinogenic, and sartans, normally whatever the sartans we are preparing, India is the biggest manufacturer of some sartans and some diabetes drugs. So we are just focusing on the you may say different nitrosamine NSA impurities. So that is also, we have a lot of applications on NSA with the help of LCMS. So as example, it's a polar LC derivation or pesticides. So we are using direct liquid chromatography, okay, in normally MPPA in water without derivation, like glyphosate, AMPA, or maybe glufosinate. So these are different polar pesticides. We have some other applications as well in the form of drugs, which is used for the veterinary concern. So veterinary drugs also plays a crucial role, because nowadays the regulatory authority body also implemented some MRL level, maximum residue limit, for those drugs, because those drugs are penetrate to that birds to get, to just for the weight gain, and those are hazardous for the human being in a certain level. So the natural, I may say, regulatory authority identified a minimum or maximum residue limit. So almost 200 veterinary drugs spiked into 10 picogram per microliter level, and 2000 MRM, multiple reaction monitoring, has been transmission with the help of both ESI positive or negative during 12-minute run. So within 12 minutes, you are able to find out almost 200 different drugs, and then not only find out the drugs, but quantification of those drugs. Now, the next one is a mycotoxins. Everybody knows about the aflatoxin, right? So it is a combination of different mycotoxins like B1, B2, E1, E2, and the mycotoxins are present in nowadays each and every food. So these are the toxins, some of the toxins, and obviously these norms are also defined by the FSSI or maybe different authority body, and those bodies basically B1, B2, or M1, M2 are some of the microtoxins we call as aflatoxins. So those toxins are present in our ready-made food or different food matrices, or maybe some of the dietary supplements, like ready-made dietary supplements are there. There are n number of dietary supplement in the market, because since last 4 to 5 years, there are different aflatoxins are also present over there. So those can be identified with the help of this analysis on LCMS. So if you see aflatoxin B1, B2, or G1, G2, even if apart from that, different vitamins like uh, vitamin B, vitamin D, how much calcium is present, what is the limit of that calcium, what are the different limits of pesticides. So those are the different applications of the LCMS. And nowadays LCMS, you may say, yeah, one more application I'm just seeing over here, it's estrogen present in the milk. So 5 kinds of estrogens present in the milk. So determination of that estrogen, we are using LCMS as a basic applications. So there are a lot of different applications apart from these. Even if the nitrofuran is one of the key metabolites whenever you are focusing on the fish matrix. So normally in the animal, some of the foreign bodies is being determination. We need to focus on its purity, and in terms of pharmaceutical industry, we need to focus the residue of a n-1 stage, or maybe some of the compound which is coming from API, some of the sample which is coming from the unknown source, and that impact on a human body. So there is a huge, you may say, stringent laws in coming last year, coming I think you may say last 3 to 4 years. So as per FDA, or as per the APEDA, or as per the different norms of the regulatory body, we need to optimize our levels, and where we are using LCMS for those techniques. So yeah, any kind of do you have any questions in your mind? Let us know on specifically on applications, or maybe anything in your mind.
Sir, it's over now?
Yeah, it's over from my side. Yeah.
Yeah, okay. So, dear participants, if you have any queries, please ask, sir, especially on the applications of the MS.
That's why I kept at least 10 minutes for the question answer. I'm expecting some questions on LCMS specifically.
Kindly ask your queries.
No questions.
Yes. There are no questions. I think actually it is hard to ask in virtual meetings like this. If it was a offline, then might have participants have asked the questions.
Okay, either session got evaporated or somewhat. What is the
No, sir, it was a
input?
nice session, especially
Okay.
on the applications part. Okay, so nobody having a question, so we will proceed. So it's a time to thank our guest speaker.
Thank you, sir.
So it was a nice and very informative session, sir. And hope all the participants are will be benefited. Actually, this thank should be a bigger one. Actually, you are guiding our participants for last 2 days. So, on behalf of Government College of Pharmacy, Karad, I express my sincere thanks to Dr. Kokate sir for delivering such a nice and informative sessions for our participants. Thank you, sir. Thank you very much. And as a token of appreciation, I would like to request Dr. Charde sir to please present a certificate of appreciation to Dr. Kokate. Kindly accept the certificate in the digital form, sir.
Thank
You'll
you.
get your
Thank you very much.
certificate through the mail.
Thank you very much.
Yes. With the kind permission, I declare the session is over now. So we will meet after the lunch break. So dear participants, join sharply at 2:30 p.m. Okay. Thank you very much.
So all participants are requested to join sharply at 2:30 p.m. Thank you.